Hello, good morning. Welcome to our webinar. We're very excited to have you here, and we will be waiting a minute or two just to let attendees trickle in. But very excited to get started shortly on our webinar topic. This time discussing what I think really gets to what drives us at Biomakers, which is how our testing can be used to actually carry over into more effective crop management. So what we'll be focusing on today is how data can be carried over into decisions and how integrated testing drives smarter crop management. Wait another minute or so for folks to join in. Looks like we have a lot trickling in now. And then we'll get started with some introductions. So yeah, just another 30 seconds or so. For those who are just hopping on, thanks for joining us. We're about to get started and really excited to talk to you today about a topic that I think really drives what we do at Biomakers into practical value for the growers, crop consultants, agronomists, and crop retailers that we work with. Really excited to be joined by some very powerful minds in the world of regenerative ag and soil biology. And I see, looks like we still have folks trickling in, so we're just going to give it a few more seconds before we get started and kick things off. But thank you all for joining today. Really excited. All right, we're a couple minutes past the hour now, so I think we're good to go ahead and get started as we have some attendees still trickling in. Today, what we'll be discussing is really the carryover from data to decisions, taking crop and soil data and actually translating that into actionable, pragmatic agronomic decisions with an emphasis on how integrated testing drives crop management. So really not just looking at individual testing, but at how things work together to develop smarter decisions and really look at systems-based management as opposed to prescription-based management. So a lot of really exciting material to get through today, and I'm really happy to be joined by some awesome folks from Tainio who are part of a very long-standing reputable company in the biological space. We'll get into introductions first and some housekeeping items. You can see our webinar agenda here. Then we'll be discussing concepts of soil health and biology. My guests will be going through some really great material on biological agriculture principles and practices and really where biology plays roles in driving crop productivity and farm profitability. We'll be discussing the different types of soil testing as well as other types of agricultural and crop testing available out there. And then lastly, we'll kind of be tying this all together with discussing how these various forms of testing fit together into a systems-based agronomic management system and talking through some examples. We have some really great data that our guests here are bringing in today, as well as some great sort of examples of how data can be integrated to drive smarter decisions, really looking at the whole of agricultural system rather than at the individual parts. So without further ado, we'll get into some introductions here. I'm Gus Plowman, Senior Technical Agronomist at Biomakers. My role at Biomakers is really focused on taking our data into practice, helping growers and other clients understand where our soil biology testing data can fit into their goals, helping achieve their objectives of improving ROI for growers, improving overall soil health, and of course, improving quality and output of the folks that they're working with across the agricultural industry. I'm very happy to be joined by two folks who I've had the privilege of hearing speak and provide some really profound insights on soil biology and agronomy. Dennis and Steve here from Tainio, who I'll hand the floor to give some quick introductions and then provide some discussion. And I will say, Dennis and Steve, you guys, what what I've heard you discuss at trade shows, conferences, and webinars has really been foundational to my understanding of soil biology and where, biologicals and biostimulants sort of fit into the grand scheme of agronomic management. And it's a pleasure to finally get a chance to share the floor and hear from you guys together, and also sort of pick your brains as we go through some of your experiences at Tainio. But for now, happy to open the floor to a quick round of intros, and then hear a bit about your philosophy at Tainio and what you provide as far as biologicals and biostimulants as part of an agronomic package. So yeah, I'll go ahead and hand it to you guys.
I'm Dennis Warnicke. I've worked with Tainio Biologicals for over 35 years. I'm the Director of Sales and Technical Support. Really, it's, I guess, the technical support part that I do. I work with growers, I work with distributors to try and utilize these tools that we're going to talk about today to find the best products, whether that be biological, nutritional, whatever it might be, to best build soil health. I mean, Bruce used to talk about if you take care of the soil, the plant will take care of itself. That's really what we try and do here at Tainio Biologicals, is help growers understand, as we'll talk about, how everything is connected and how we can put those pieces together and move forward.
Yeah, and Gus, thank you and Biomakers for having us be part of it. We've been listening to your presentations, and I know that I've pulled some great insights and I may have borrowed a few ideas that you have presented on, because it's great. It's been fun working together and bouncing questions back and forth, because it is a very interesting age that we're living in, where all of a sudden we're starting to have the ability to collect data that can really drive some of the decisions. And that's kind of what we're talking about. I'm Steve Becker, Chief Science Officer at Tainio. I work with some of the growers, distributors, formulators, but a lot of what I do is helping maintain our biological library, I guess is the best way to put it, and keep all the microbes happy.
Yeah, and the exciting part about this, just one more thing, Gus, is the fact that 30 years ago, we talked about we could do soil testing, we could do tissue testing, and then there was plant sap testing, but we really never had a good tool for biological testing. And that's what's so exciting about this, is now we have those tools available to really start utilizing and implementing into agricultural practices as we move forward.
Mm-hmm.
Exactly. Yeah, a lot. That's exactly what we'll be getting into today. How to go about testing rather than guessing, and what tools factor into that equation. One last sort of housekeeping item that I forgot and glossed over and jumped right into intros. But before we jump into an introduction to Tainio and your principles, I just wanted to mention to folks in the audience that you can feel free to send in your questions. You'll notice a chat box. We're going to be going through some very technical concepts today, so we want to make sure we leave maybe 15 minutes or so at the end of this webinar to answer your questions verbally. And for anyone who, if we have too many questions that we don't get time to answer, we will follow up with you after the fact over email to ensure that we get all of your questions answered. So don't hesitate to reach out with those questions, and we'll try to field as many as we can on the back end. So anyways, happy to hand it back to you guys and go ahead with, we'll go through kind of top to bottom, where, from a conceptual framework down to the nitty gritty practical boots on the ground applications. But yeah, Dennis and Steve, feel free to take the floor.
You got to start off with a bad joke.
Well, actually it's pretty funny, because I love this. I love the far side. Yeah.
It's not bad.
And, and he gives it to me because at my age, I can talk about this, because
I grew up with far side.
Yeah. Hey, you little softies, when I was your age, I had to crawl through 14 inches to the soil surface and back. That's
uphill both ways.
Uphill both ways in a snowstorm.
Oh yeah.
But actually, man, despite his artistic pretensions, his sophistication, and his many accomplishments, we owe our existence to a 6-inch layer of topsoil and the fact that it rains. And that's really what it comes down to. And that's what we talk about here today, is what can we do actually to build that 6-inch layer of topsoil? Because when we start to talk the fact that it rains, water holding capability, everything is dependent on that 6-inch layer of topsoil. And how we grow that is critical to biological life, to plant life, to root development, all of those things.
And that zone, that skin of the earth should be full of life. It's like our skin. If we lose that protective layer, I I know I've seen movies where people lose their skin. It gets real weird.
Yeah.
Things start to fall apart, and the same thing kind of happens with our land and our soil. If we start degrading our soil and turning it into dirt, I know we've all heard that silly one. Don't treat your soil like dirt. But that's really what we need to focus on, is building life back into these systems, because for so many decades, conventional ag, and I don't want to point fingers too much, I'll point them a little bit, but so much emphasis has been placed on how can we kind of guide and force nature into what we expect. And now we're kind of stepping back and realizing that it's the biology, it's the life that's driving these systems. So that's where we can start to have a better impact and have a better way of improving some of these systems.
It's all about growing soil.
It is.
That's what we're doing. We're farming soil.
Yeah. And it's a succession that we start to see, if you want to move on to the next one. And that's kind of what we're seeing is we start off with the very beginning bare rock. The first organisms that are there are known as chemolithotrophs. They're bacteria that digest rock. So they're the first ones that are pulling off little tiny bits of nutrition so they can slowly get this digestive system up and running. And what we're looking at is we're increasing complexity. We start off with very few, very simple organisms. We start add some lichen, and then we get into our small annual plants. And that's usually where we start to see a lot of our weed species hanging out and living in that area. And as we start to increase our complexity, we get into more shrubs and trees, and then eventually our kind of climax or apex communities, shade-tolerant trees, we've got deciduous trees. We've got a very complex community that is full of bacteria, fungi, protozoa, microarthropods, you name it, they're all there. But it's this idea that we're increasing complexity is what we should be doing, but we can also do the opposite.
Well, and that's a lot of times what I see in agricultural practices from my experience is rather than moving forward, we're moving backwards. Yeah. And that's where all this testing, the things that we're going to get into today and talk about is how do these tools, these testing tools help us guide us to move forward in the best way we possibly can. And, I mean, we kind of look at, the foundation is being bacteria, but identifying this, it comes down to being able to test that within our soil environment. And that's some of the things that we do.
Want to move to the next one?
Yeah.
Perfect.
And yeah, so, I mean, you can really speak about this. John Kemp talked one of the best ways to build a good fungal community is through beneficial bacteria. Yep. And the reason for that is that's the foundation. That's the food. That's where everything starts, and we build from there up. And before, we didn't really have any way to measure this, but now there are tools out there, and even actually from what Biomakers doing now is we can see that. We can get a test and we can see our bacteria to fungal and what things we need to do. And so, I mean, this is some research done by Montana State University. By just adding bacteria to the soil environment, we see an increase in fungal community.
Yes.
Not only population, but also diversity. And the exciting thing I get on that is the idea that I call it habitat. We're building habitat in order to support that, like you say, bacteria, fungal, protozoa, that stair-step ladder that Dr. Hatfield talks about, moving up that ladder to soil health.
And humification is the process where we're actually building soil, and that requires the bacteria, the fungi, it requires those organisms that are down there digesting, processing, and working those carbon molecules back together to create this structure, to create this skin. So we can be building this skin, we can be creating this skin, or the converse of that is we can be degrading and destroying. And that tends to, like we looked at the succession, we can either be building that complexity to higher quality, or we can be degrading it and generally encouraging different weed species to grow up. And that's because we kind of consider everything to be interconnected.
Yeah, and that's, I mean, Bruce used to talk about this natural growing through biology. Everything in the universe is connected to everything else.
Next slide.
Go ahead to the next slide, Gus. It's all connected. You cannot isolate one mineral, one soil, one microbe, one plant, one animal, human component, and treat that component as the others don't exist. Once you get an understanding of the interconnectedness of all things. And I talk about this with growers all the time. We plant a cover crop, and then we go out and maybe we spray it with a herbicide, we till it back into the soil. Well, these three things are all connected. How far did we move that needle or that stair step to that next level of biological health or soil health? And so, you know, you gave a good example the other day. I always say it's the missing piece or the missing pieces. What are the missing pieces of the puzzle? And we talked yesterday, Stephen, you brought up a good example. If you have one person putting a puzzle together, it takes time. It's not as quick as if you have 6 people working together that build that outer perimeter of the puzzle and put all the pieces inside. And that's really what we talk about. When you start to add the plant, the microbe, the soil, the human component, all of these things together, and you have the tools to test them, which we're going to get to, now we can really start building soil health.
A holistic approach versus a reductionist approach.
Yeah.
And that's where we, no, you can go ahead and move to the next one. And that gives us, again, we've mentioned this idea of that outer layer, this living skin, and that's what we're trying to build. That 6 inches is where all of our money is made. It's where all of our food is made. It's where all of this life really needs to be focused. And that's where our nutrition is coming from. And it's that nutrition that's leading to maximize photosynthesis. And maximizing photosynthesis is how we get better exudation, which feeds those microbial communities, which builds that soil. It's this beautiful cycle. It's almost like nature, you know, kind of did this on purpose. It's weird.
Yeah.
Almost makes sense.
Yeah, because plants, animals, humans, we all need 5 things. We need light, we need heat, we need air, we need nutrient, and we need water. We need all those things. And with the exception of light, the soil actually can supply each one of these things. You know, we talk about temperature, soil temperatures. We see it all the time with good microbial activity within that soil environment. The temperatures are higher. We see air, oxygen, polysaccharides, building soil, lack of compaction. We see nutrient availability, which we'll talk a little bit here in a minute, and water holding capability. All of these are tied together, but if all of these are not in balance, we'll just use the word balance, then we cannot have optimum soil or plant health. You know, it goes down to the law of minimum. I mean, that's what we talk about all the time when we focus on deficiencies being our biggest limiting factor.
Historically, that's what we've been focused on.
But we can have a deficiency in soil health based off of light, heat, air, temperature, water. We see all the time, compaction is our problem. We don't have enough water going through the growing season. All of these also have to be in balance. So when we start to talk about the law of minimum, we have to have soil into the state of this barrel. We have to have all of those components. And I always say that an excess by something that we apply causes a greater deficiency than, I mean, if there's a deficiency in that soil environment, that's easy. You go out with a soil or a foliar application, or you use a microbial inoculum in order to make that more available within the soil environment. But if we have an excess, now we have to overcome that excess. We have to stop what we're doing. And the only way to do that is understanding through testing of what we're actually doing. And that's one of the things, like when we start to talk about soil health and the previous slide, you know, utilizing the BeCrop test gives us that information of how do we build some of those limiting factors. It's, I mean, it's an exciting time as far as I can see it, because really it comes down to you are what you eat.
Yeah, and that's the next slide if you wanted to move over. And this is something that I think Gus and we have, using human health as a comparison, as an analogy to what's going on in the soil, it makes sense. When you start looking at the plant's root, when you start looking at the plant root and you see all those little tiny hairs, and then you start looking at our intestinal lining, they're very similar structures. It's this idea of increasing surface area, increasing the total area that we can have different microbes and biology surviving in that area. And it's because it's the microbes that are good at extracting the nutrition off of that soil. Like we were talking about initially from bare rock, those chemolithotrophs, using their enzymes, using their acids to break off those small individual, basically particles of nutrition. That's what's going on. That's what's going around on in our gut, as well as the plant gut. And the plant gut is basically just the microbes in that rhizosphere, that rhizosheath, those microbes right around it. And we see basically the same idea. We see similarities in the necessity of different nutrients for a plant's health as well as our health. And we like to say, I mean, we've definitely been using Dr. White's research. And if you're not familiar with Dr. White's research on rhizophagy cycle, don't stop right now and go check it out, but very soon. Look it up, check it out, because it is incredible. It is this entire process explained. Back in the 80s, Bruce Tainio, the founder of Tainio, he had this understanding, and he was focusing on, okay, to maintain plant health, it's all based on nutrition, and a lot of that is trace mineral nutrition. How does the plant get those trace minerals? Well, he understood that it was through the microbes, but we didn't really understand how that was happening. It wasn't until 2010, 2011, and 2018 with Dr. White looking at this rhizophagy cycle that we really started to understand this is purposeful. This is plants physically eating microbes. They're encouraging those microbes, they're feeding those microbes with their exudates, and then we have this the root meristem, this area I can't point to it easily, down at the tip, where all these microbes are being basically encouraged to grow based on those exudates. They're pulled into the plant, they're bombarded with reactive oxygen species, superoxide, which breaks the microbes down, and the plant can then digest some of those microbes. It's physically eating some of those microbes. The ones that it doesn't eat, a lot of times it's cloning. It's creating duplicates of those because it knows that it needs those organisms. Then it spits them right back out. It's extracting nutrition from them. They're really hungry. They go back out in the soil. They go back to mining that soil, extracting that nutrition, and it's a cycle. It goes round and round. And the research is showing that the plant can purposefully select the organisms that it wants and it needs. If it needs more phosphorus, it's going to increase the phosphate solubilizers. If it needs more potassium mobilizers. If it needs more nitrogen fixers. And so this idea of, I like to think about, if you've seen pictures of microbes, they kind of look like little pills. And I think about them as basically, these are the little pills, these are the little food packets for the plant. In most biology, it's roughly a 10-2 plus everything else. And that's the beauty of a plant eating through rhizophagy, is just like you and I are made up of all of these nutrients, the microbial body is the same. It requires all of these things. If I go out and apply potassium chloride, if I go out and apply anhydrous ammonia, I am hitting one thing. And like Dennis was saying, an excess can oftentimes lead to deficiencies and other things. When we're trying to force feed our plants, we end up with massive imbalances, and it happens very quickly. The converse is if we allow the bacteria and the fungi and microprotozoa and all these other organisms through the rhizophagy to feed the plant is selecting who it needs, who it wants, it's extracting nutrition, it's cloning the additional organisms that it wants to be in that environment, and it's feeding itself, and it's getting a balanced diet.
Well, yeah, so basically the plant is farming microbes.
It is.
I mean, that's 100%. Really what it comes down to. And that's really what we want to tech to. How do we get that plant? What tools do we have to get that plant functioning at the highest level based off of nutrition, photosynthesis, yes, microbial, the rhizophagy? How do we get all these things? How do we as growers not interrupt that process? And that's really where the testing comes in, because yes, we've always said, test, don't guess. I mean, that's really what it's come down to. And, you know, we use soil analysis, again, to see what is in that soil environment so that we don't create an excess. We know what we need to put out, but we also use plant sap analysis to see what is available in that soil. You know, we used to utilize those based off, we know there's phosphorus in that soil environment, but the plant's deficient based off of soil. So we want to use organisms that can help make phosphorus available. And, you know, we utilize all these tools, and now we have the Biomakers tool, which gives us that next step of understanding what we need to do. So we had
soil, we had plants, we were kind of missing that interchange, and that's where this idea of the microbe functions.
The piece of puzzle, the missing link.
And the missing piece.
You know, that's, I mean, jump in here. I mean, we've actually dominated most of this conversation. I feel bad. Jump in here and talk about, you know, some of the things based off of the testing and what you see.
Yeah, thank you guys. No, I didn't want to interrupt because this was all such great material and conversation, and to kind of jump back to what you were explaining about the rhizophagy cycle, about these roles microbes play in the soil, you know, solubilizing nutrients like phosphorus and potassium, you know, mineralizing and unlocking nitrogen, you know, converting organic nitrogen to ammonia and nitrate for the crop. We've had a lot of folks who, you know, and not to point fingers at conventional ag, but the mentality the last 20, 30, 40 plus years, since chemical and physical soil testing have historically been what's available. You know, these two boxes on the left looking at soil texture, water holding capacity, bulk density, and then our chemical extractions have really been the tools that have been driving fertility applications. The Malik full panel tests and other phosphorus extractions like the Bray extraction out there are, they're great tools to see sort of what's chemically available. But what we have a lot of folks coming to us at Biomakers because what they're seeing is that their chemical tests, they're seeing that they have plenty of, high levels of nutrients in their soil, but then they're running into deficiencies, yield drags, plateauing or declining yields. And that's where this section on the right, the other tests here, plant tissue and sap is another big picture, because that's where, in an ideal world, and historically what conventional agronomy has assumed is that if you augment the level of, say, phosphorus on your Malik test, you're going to get a response of phosphorus in your plant tissue test. But in my experience at Biomakers, so many folks who have come to us have really, looked into biology because they're seeing that their tissue test is not showing that response to phosphorus input that they're seeing on their Malik. So they're wondering, what's the missing piece here? And that's where the biological soil testing comes in. As you guys mentioned, really the newest sort of tool in the toolkit to see why there may be nutritional uptake issues despite having a lot or even excessive, fertilizer or nutrients in the soil from historic heavy fertilizer applications. And what I think is really driving people to think about the biology and use tools like our testing to look at this is that there's been a lot of studies in recent years. I know there was a big landmark study a year or two ago that showed that plants, or I think it was looking at corn and other major row crops, they do not obtain most of their nitrogen from fertilizer inputs. They actually obtain upwards of 60+% of it from the biology in the soil. The researchers did this by essentially labeling the nitrogen that was being applied and then analyzing the plant tissue. And they found that only maybe, I think it was in the range of 20 to 40% of the fertilizer that was applied on their field was actually making it into the crop tissue. The rest of that, the majority of that nitrogen is actually coming from the biology. So that's why growers are coming to us with, their Malik looks good, their tissues are showing deficiency. And then when they run a BeCrop test and look at the biology, often they see very deficient levels of these phosphorus cycling, potassium cycling, nitrogen fixers. And of course, that's where, if they apply more nitrogen and phosphorus, they're really not getting at the issue. They may even be exacerbating the issue with imbalances, as you referenced, Dennis. And when they run a BeCrop test, they can see, okay, maybe the issue isn't that I don't have enough phosphorus, it's that I don't have enough phosphorus cycling biology. And of course, that's where, products offered by companies like Tainio can really fill that gap where you can, if you apply a biostimulant, you may get that response of, phosphorus solubilizing biostimulant like Tainio offers can help get that phosphorus available for the crop so that we address that deficiency rather than, essentially just banging our heads against the wall and applying more and more, expensive phosphate inputs year over year. With BeCrop Technology, we can really diagnose those biological issues. And of course, I did want to shout out, the Haney test is a big component of, we offer it as well at Biomakers through our partnership lab, Regen Ag Lab. And you notice the Haney test actually spans both the chemical and biological categories here because it does have some nutrient extractions to look at both available and unavailable nutrients for the crop. And then it also looks at biology through soil respiration. We really like the Haney test because it's a great way of sort of complementing the BeCrop test of what's available to the crop, what nutrients aren't available, and then the BeCrop test tells you, okay, who are those players in the cycling process that can really get that, be important to make those nutrients plant available. So really quick, I wanted to do a quick introduction to BeCrop Technology and what we, the services and testing that we offer at Biomakers. So for those who are unfamiliar or maybe joining us for the first time, Biomakers, what our team has done over the last 7 or 8 years or so is develop the largest soil database focused on agricultural soils, currently over 24 million microorganisms and growing. So we've really looked across, we have this large taxonomic database of these millions of bacteria, fungi, protists and other microorganisms we can identify. And we've not only, developed the tools to identify which microbes are there, what their scientific name is and their abundance, but also computing technology that profiles the functions of those microbes. And that's ultimately what's important for agronomic insights, because not even, not even a well-trained soil biologist is going to know what thousands of different, what specific functions millions of different microbes are performing. But with the bioinformatics processes that our data science team has developed, we're able to see the levels of phosphorus solubilizers, nitrogen fixers, these other plant growth promoting bacteria and fungi that we've referenced. We've carried over a lot of our research into peer-reviewed publications, both by us internally and by some of our collaborators, just to validate our technology and ensure that it's scoped out by the peer review research community. And we're a global company offering the testing across 6 continents and 50 plus countries. So that's just a quick blurb on Biomakers and our technology. And now, of course, the more exciting part is what we actually provide on the BeCrop test. And for the sake of time, I'm gonna try to go through this pretty quickly. But basically, the parameters that we provide break down into 3 main categories: soil quality, which is really looking at the whole community. These metrics include biodiversity, which is very similar to the metric that we were looking at on those bar graphs from Tainio of how the Spectrum product improves fungal diversity. So we look at the diversity of those organisms, which obviously is a great proxy or indicator of soil health. We also look at the functionality of those microbes. So whereas biodiversity, of course, is based on how many different species there are and how prevalent and diverse the populations are, functionality doesn't look at the species taxonomy, but looks at the functions those microbes perform, because one beneficial microbe may perform many functions, other microbes may perform very few functions relevant to crop productivity. And we also look at resilience. So that's really the adaptability of those microbes, or their ability to deal with stressors like drought, tillage, fungicides, high salinity. And then, of course, these are some really common biological indicators we also include that are offered on other testing, like phospholipid fatty acid. We include a fungi to bacteria ratio and a mycorrhizal ratio, which are also sort of benchmark indicators of overall soil health. So soil quality is very overarching. We're looking at the whole population when we calculate these metrics, but then our other 2 categories, plant health and then nutrient cycling, this is where we really get down into the specific functional groups that are relevant to agronomy. So this is more where the specific agronomic insights and some of the quick use case that we'll be going over in a minute comes from. Plant health includes soil-borne diseases that we're able to diagnose, biocontrol agents, microbes that produce phytohormones or beneficial metabolites that aid in healthy plant growth and root growth, and then also stress adaptation. We look at microbes that are able to help plants deal with drought, soil salinity, heavy metals, and other stressors that crops will encounter in the field. And then lastly, nutrient cycling, of course, pretty self-explanatory, is where we're able to quantify the microbes that cycle and unlock nitrogen, phosphorus, potassium. Carbon is a big component of what we look at. So we look at microbes that fix and sequester carbon, as well as microbes responsible for breaking down organic matter and crop residues. And then last, we also look at micronutrients, not the micronutrients themselves, but of course, the microbes that are able to cycle and unlock micronutrients, make them available for plant uptake. And then, as I mentioned, we have additional chemical fertility test offerings to, we don't just want to focus on the biology. We know that the biology is a missing component, but of course, the historic components of chemical and physical soil traits are also an important aspect of holistic management. So these on the bottom here are some of the examples of chemical fertility tests that we offer in addition to our services. And I did want to hand it back over to you, Dennis. I think you had a sort of a quick anecdote related to some of the parameters here, specifically in the disease side, that I wanted to make sure I don't gloss over and I give you a chance to touch on.
Yeah, I would have interrupted you guys. Anytime I have a chance to talk, I would have cut right in. No, it's actually really interesting because, as I said, I do a lot of technical support, utilizing information to help growers make educated decisions on what they can use. And when we were doing a trial, and we were testing the control using plant sap, using soil analysis, and using the BeCrop in order to identify what's going on as a learning tool and as an educational tool to this biofarming group to understand better what's going on in their soil environment. The cool part about it, in April, I pulled a test. I pulled a bunch of BeCrop tests. And what's interesting when you start to talk about soil-borne diseases is I pulled the test. Based off the test, the health was very low. It had a very low BeCrop score, But the interesting thing, it gave a whole list of diseases that wheat was susceptible to. And one of them was stripe. And I took this test back to the grower and I said, look, you guys are very susceptible.
It's here.
And he said, funny thing about that is they said, when you pulled that test, we didn't have any. And yet that has shown up. We had to spray a fungicide on our control and the rest of our field because of exactly what you just told me was going to happened based off of this information. And so when you start to look at these tools and the information that they can give you, and just the decisions that can be made based off of all the information in here. You know, you talk about the nutrient cycling, which is part of overall plant health. Getting into those secondary plant metabolites so they can resist it. When we start to talk about silicon, calcium, and all of those things to build cell wall strength, all of these are part of this huge picture. But it was pretty cool to pull the tests, see the information, and have it express itself, all based off this test in a relatively short period of time.
Yeah, and anytime we can look to the future, basically, why would we not? It gives us one more tool to have a better understanding of what's coming up. What do I need to prepare for? And if you can prepare for something, why would you not? It allows you to go through that, because anytime we have any of those stresses, every bit of stress the plant undergoes, it reduces yield. Every time, I think about it like a balloon, a water balloon. And every time we have a stress, it's like poking a little tiny hole and it's leaking out until the very end of the season, where you might end up with that as your yield, or you might still have a pretty full balloon. So if we can get ahead of these things and know that they're there, it gives us a chance to focus on them and to address them before they're even a problem.
Well, and your test shows, you know, as we always say, the diseases, soil-borne diseases are there.
Yeah.
Whether they express themselves is basically environment dictates expression.
Yes.
And so with this test, it shows whether they're there or not, but it shows susceptibility also. You know, that when you start to think about that, that's like going to a doctor and getting a blood test, and knowing what's going on in your body. Now we're knowing what's going on in our soil, our plant. So we'll give it back to you guys. I just thought this was pretty cool and relevant here to what we're talking about. This is just one way we use this tool. There's multiple other ways when we talk about all of these other things you talked about, but you know, we're running out of time. We're taking too much of your time. Let's move on.
No, thank you guys. I really love that anecdote, and I appreciate you sharing. I think, you know, that's exactly what our disease risk indexes are intended to be, sort of an early warning signal. What diseases are there? What is their risk level? And it's not uncommon for us to have that scenario where growers say, oh, it looks like I have a disease, but it hasn't manifested itself. And then, you know, a few weeks or months go by, they see that disease, you know, manifesting in the crop. And a lot of folks who have been using our testing as a diagnostic tool, they're using that early warning sign to determine their crop protection product program. And it really gives an extra angle, because when I was running, you know, before joining Biomakers, was running organic crop trials, and that was particularly challenging, especially organic crops, specialty crops in the Southeast, because we have so many pathogens, so much humidity and heat down here. And a lot of times, this was before this was several years back before I was working with Biomakers, but a lot of the, when we ran into a disease issue, it was almost always a post-mortem analysis where we'd see a disease popping up and we'd take plant tissue samples, you know, uproot the crop from the really infected areas, send it to our diagnostic lab, get a diagnosis 4 days later. And by then the disease is already beginning to spread. And it's pretty much at that point, really a losing battle to try to fight it. But getting that warning sign, that indication that you have a disease that may potentially manifest itself weeks or months in advance, or even in, even the season ahead of time, really gives an additional sort of value as far as figuring out what you're going to do as far as IPM and crop protection products, how to best manage that problem before it arises and becomes a concern.
Yeah, before we have to go down the aisle of death to pull out one of the nasties, we can utilize those beneficial tools in order to help our crop, which is not only going to help the crop, the yield, the soil, the microbes, all of those. Rather than have a negative effect, it all has a positive effect with that information and utilizing that tool.
Yeah, and I really like that what you alluded to and have a couple times so far is that this is all interconnected. You know, disease doesn't just pop up. I mean, disease is very sporadic, but at the end of the day, it's driven by environmental parameters, plant stress and susceptibility. You know, we actually see that with our stress adaptation metrics we look at, that we have had a few projects. I remember one in potatoes where we saw the disease was very present, but the client actually shared with us disease scouting data, and we found that where the disease was less impactful was not so much based on the presence of the disease. The disease was everywhere, but it was actually the beneficial microbes that provide these stress adaptation metabolites, basically secondary metabolites that help the crop prime its defenses to pathogens. And the disease impact actually correlated more with that. We've also, I think that, you know, other folks have done a lot of work on how crop fertility and nutrient cycling affects disease susceptibility. And that's our use case here is going to get into some nutrient related indexes. But I do want to mention that if you can use our testing to hone in and really, you know, make sure that biological nutrient cycling is on point, then you may have a disease present, but it may not rear its head as significantly or be as big of a risk if the crop has access to those vital nutrients. You know, you mentioned silica, and there's, you know, zinc can play a vital role in disease response, other microbes especially. So it's all connected, but of course, from a diagnostic approach, you got to think about the life cycle of the plant, the relationship with the soil, and that's what we're going to get into here with a quick use case and some biological soil data, some BeCrop data from Tainio's actual tests. So with an integrated testing approach, as I just mentioned, we're not just taking a look at crop tissue tests, seeing a deficiency, and then going out and applying a fertilizer or foliar. We're also taking into account what's there in the soil. So soil chemistry tests like your standard Malick, or ideally, you know, your Haney test that gives you an idea of the potential root cause of chemical fertility issues. And then the biological component really is another angle to look at those potential root causes, because it may be that you have plenty of phosphorus in a soil, but you're getting a deficiency in a crop because you don't have those phosphorus solubilizers or other nutrient cyclers. So we're going to walk through a real sort of case study of that, and also get into some data that I know you guys have from your wheat trial. So I'll hand it back to you to just kind of go over the scope of this trial and some of the outcomes that you saw.
Yeah, and this is just another form of testing. We were doing a wheat trial, and we were originally, this was based off of 13 different varieties of wheat and trying to identify which varieties reacted differently to different fertilizer applications to try and find a variety. Obviously, you know, Bruce was a seed geneticist, used to breed wheat. Whatever you breed that wheat for is kind of what it's going to like. So if you breed it for high nitrogen, it's going to want high nitrogen. And so we utilize some of these trials to try and find better varieties to reduce the utilization of nitrogen, as you talked about, utilizing those bacteria, that nitrogen fixation, which we won't even get into, in order to supplement that nitrogen deficiency, at least perceived by us as growers of how much nitrogen do we need to grow a bushel of wheat. So as you can see, the standard check was 120 pounds, one-time pre-plant application. We had another one which was 120 early, and then a top dress of 34 more pounds in April. And then the third was a split application, spring and fall, of 120 pounds. On the biological side, it was a little over 25 pounds, 25 pounds of nitrogen plus a little bit of fish.
A little bit from the fish.
Yeah, so a little over 25 pounds of nitrogen. That's kind of why we refer to it at. But the amazing part about this, when you start to look at yield, when you see across the board where all of these yields are within either a little bit better or a little bit worse, but we're using 1/5 or even less than that of the amount of nitrogen used in this trial to try and grow yields. And when we start to talk about that, when we start to talk about the cost of nitrogen, we start to talk about building soil health, not burning up that carbon, that humus within that soil environment, you know, it all really starts to make sense. And, you know, we can kind of, I mean, I just wanted to kind of look at this because it is possible. And what's really exciting about this is not only did we do soil analysis, plant sap, and these were the programs, and we looked at cost, and we're looking at ROI, we're looking at all of those things, but we also did some plotting based off of the Biomakers testing. And this is what we got really excited about when you start to see that expression within that soil environment of what's going on based off of some of the things that we talked about.
Yeah, some of the ways that we can use the tool. And like Gus was talking about resilience, this idea of adaptability. How many organisms are there doing the same job? If your team's missing the quarterback, it falls apart. If you've got backup quarterbacks, if you've got other people that can do the same job, That's where this resiliency idea comes in. And when we see that we're applying more nitrogen, it shuts down rhizophagy, it shuts down the plant's need for those organisms. If the plant doesn't need them, it doesn't feed them, it doesn't have to have as many. The unfortunate side of that is, like Gus was saying, they do lots of things. So if we're missing some of these organisms, now we're missing some of their other capacities. And that's where we saw when we have lower nitrogen application, we have a higher species diversity, we end up with a better resilience. That plant is able to adapt to stress and survive through these environments better. That was one of the interesting things. So we just pulled out a few bits of data. There's so much information that you can get from the BeCrop testing. We also saw some calcium, we saw magnesium shifts and changes. We saw, there's the calcium. And here we have phosphorus shifts and changes as we have more of those organisms making more phosphorus available. Well, it feeds the plant.
When you see plus 15, I mean, that may not mean a lot to a lot of people, but that's statistically significant when we start to talk about this based off these tests. I mean, and you know, in a wheat trial, everything's very close together. So to see these significant shifts based off, again, all of these functioning of all of these pieces that we put together, the biological, the nutrition, all of these things are working together. And we start to see in the soil environment how quickly we can shift that biological community based off a habitat and environment.
Yeah. And the secondary benefits that we can see associated with that. It's not just one thing. It's this combination. It's this interconnectedness. As we increase the biological diversity, we increase the soil's capacity to feed the plant, protect the plant, build soil, build gas exchange, build moisture holding capacity. All of this comes together. Or again, the converse of that is we degrade and decline all of that, and we reverse our succession and go to a more simple digestive system, a more simple soil that ends up with more problems generally.
Did you have something you wanted to say, Gus?
Yeah, I was just going to give a little bit of context to what you guys covered it great. And, you can see pretty easily across these charts, the bottom cluster, the Tainio product, I believe it was Spectrum, if I'm not mistaken, or maybe it was a whole program that you were looking at in the sweet trial. But basically you can see that purple, the Tainio program was higher in each of these cases. And for those who have not worked with BeCrop tests, basically the way that this scoring plot works is we have a 0 to 100 index. Basically, it's like a percentile scale where we score the levels of these microbes and these other indexes that we look at based on the microbial functions. And then we can see the BCS or BeCrop score, as we refer to it, is that 0 to 100 index. And we also have, I know it's a little bit blurry, but there's indications here of which ranges are very low, medium, high, and very high. And obviously for most of these metrics like resilience, nutrient cycling, the growers we talk to who are long-time regenerative cover crop folks who have been doing no-till for decades, a lot of them who've really been invested in soil health for a long time tend to fall into more of the medium to high ranges. And that's what we like to see. And those are the folks who are really getting good ROI, not using much, as much synthetic fertilizer, maybe using a little, but not using massive amounts. And then the folks who've been long-term, using long-term intensive tillage, ramping up their fertilizer applications year over year, more costs, more input. As Dennis and Steve have mentioned, as you ramp up those inputs and the system becomes more dependent on those synthetics, we can see a decline in those nutrient cyclers. So with BeCrop, and this tool is available for free through our portal to compare samples. You can see the differences. What really stood out to me in this trial was this resilience index. I mean, a 46-point shift is almost, think of that like 46 percentile. It went over almost half, it increased by almost half of the soil samples ranked in our database here, from the very low range, a score of 19, all the way up to the high range with an average of 65. So big, as Steve was saying, basically the soil got tougher, more resilient. You have more adaptability of those microbes, so you're more likely to get their benefits rather than lose their benefits when they do encounter some stress or when temperatures go down or moisture goes down. And then nutrient cycling, the rest of these indexes we looked at, these are all anything over a 10, even a 10 percentile or 10-point shift is notable. I would say, just like you might see, a 10% shift in, overall phosphorus or pounds per acre, or potassium, or whatever the case may be, can be meaningful. And in our index, a 10 percentile change can be pretty meaningful. A nearly 30 percentile change from about the middle of our ranking, 52 to 80, is quite notable. But across potassium, and then magnesium, calcium, and then lastly phosphorus, we all saw increases of greater than 10 in this particular project. And that's showing that really, the Tainio program is doing what it's intended. You're able to get feedback and ensure that you've diagnosed the issue, but also you're addressing it and seeing the needle move in the right direction. You're seeing those populations of phosphorus and potassium solubilizers build and increase relative to the check or control. And I know we're getting up on time, but wanted to run through a quick use case. Yeah, I tend to get a little long-winded when I get into these.
That wasn't you, Gus.
It's you get excited. It's fun.
Yeah, it's exciting stuff to nerd out on. But anyways, a quick allusion here to your leaf extract data, which I'll let you guys comment on, but sort of is that, ultimate out really seeing what the results of these improved populations of nutrient cyclers are.
Well, the amazing part about this is, again, another test to identify all of those things that we saw happening based off the BeCrop test, and then pulling a plant sat based off of plant expression. The amazing part about this, again, it was 120 to 154 pounds of nitrogen applied in the conventional. The test that we have here was the 120, just so you know.
Yeah.
On our side, it was a little more than 25 pounds. If you look at total nitrogen, the total nitrogen in the biological is higher than in the applied.
Yep.
So the nitrogen was higher, the potassium, the magnesium, the calcium, all that we talked about did better on the BeCrop score. The manganese, cobalt, phosphorus, silica, iodine, boron, molybdenum, and selenium were all higher in the biological side. And this, I use these tools. Now we see that's based off of, we gathered information based what's on the soil, what's in the plant, and what's happening with the BeCrop test based off of digestion. Because a lot of times you make your biological choice or recommendation based off of all of this information.
All those pieces coming together.
Exactly. And that brings us, that's a perfect segue into our integrated testing case study here. So this is a generalist example. So we're taking data here that's a little bit simplified to show the process of integrated testing. So this is not necessarily real data, but this is a real scenario that we've gone through with countless clients, and especially the clients who are the growers who are really honed in. They really want to get to what that yield limiting factor is. This is often sort of what they're already doing before they come to us, but this is how the BeCrop test can add another layer to how they're tracking those soil plant nutrient relationships. And basically in this line graph, what we're seeing, we have two axes here. On the left, we have our plant tissue and soil fertility in ppm. And then on the right, we have the BeCrop score index. And initially, in this scenario, this is about a roughly, sort of around 2 season study. And initially, we can see in the brown, we have the soil phosphorus, the fertility based on a chemical fertility test, and then we have the leaf tissue data. And what we're seeing is a discrepancy here with that leaf tissue data. And this is, if we're looking at phosphorus in a sap or a plant extract test, ideally, I know from some labs, they say they want it to be around 140, 150. So the leaf tissue is very deficient, but 60 ppm is actually very high for a soil fertility test. That's somewhere in the ballpark, I know, of 100 pounds per acre. So as the leaf tissue is very deficient, and from the first sample taken in around harvest time of 2021 was a concern. So conventional ag standpoint, a grower would probably apply more phosphorus to try to address that leaf tissue issue. So we may see their soil fertility phosphorus go up here. But then going into an early season sample in around May or June, they're seeing that leaf tissue phosphorus level drop even more. So that deficiency is even exacerbated. So this is often when growers come to us, come to agronomists and crop consultants who have a biological background like Tainio, and they often will run, this is where a BeCrop test can offer another layer to why we're seeing this discrepancy between soil fertility and plant fertility or leaf tissue analysis. And here they take a BeCrop test, and you can see that in the blue we have the soil biology phosphorus level and the BeCrop score index here, which ideally we want that to the phosphorus and the other nutrient cycling indexes, we want to see them in the 50, 60 plus range. At least that's about average. But in this case, it was all the way down around 10 or 12. So it was very deficient, very low. And that's likely what's leading to this discrepancy and this nutrient uptake issue, is that they don't have the biology there to unlock that phosphorus. So here's where they can take that corrective action. They take a BeCrop test, diagnose the issue. It's, poor biological phosphorus mobilization, and they can apply a product like Spectrum or another product that, experts like Tainio can tailor to that problem. So, the folks who've been in the biological product space for a while know which species and products are tailored to inoculate phosphorus solubilizers or other microbes that may be needed. And then ideally, as you apply that product, like we just saw in the data on the previous slides, can augment that biological phosphorus cycling level. And we see that increase. Ideally, as those microbes are getting established and unlocking more phosphorus, we see the plant uptake increase, as we see an uptick here from their early to their mid-season tissue tests. And then as time goes on, depending especially on the recommendations of the soil inoculant or biostimulant expert, as they continue with a biological focused program, ideally we see that biological phosphorus data from the BeCrop score continue to increase over time. And as that increases, we see the soil the leaf tissue phosphorus levels increase to getting close to that level we want to see of optimal in the crop. And then we may even see a little bit of a decline in the soil fertility as the crop is uptaking that microbes are churning out phosphorus, the crop is uptaking it. That brown line here may decline, which if you have an excess like 60 ppms of phosphorus in the soil, that can help bounce things out and start to move really by the last time point here. This is the direction that we want to be moving to address those yield limiting factors. So we're running through time, and I want to make sure we have a few minutes for questions. But as a last little tidbit, I wanted to shout out the new service that we're offering at Biomakers, BeCrop Farm. Historically, we've provided testing on a sample by sample basis, but of course we know that can become very prohibitive to cover thousands of acres when you want to pinpoint biological deficiencies, not just in one field or one setting, but across large-scale row crop acreage. So with BeCrop Farm, what our team has developed is an ability to take satellite-based environmental data and use that to fill in the gaps as far as soil biology data. So here we have an example, a demo of BeCrop Farm, where testing is done in specific sites. So we receive the field boundaries or maps of fields from our clients, and then we run a environmental data check that looks at the map in satellite-based imagery and identifies where to best sample to create an accurate biological map of the fields. And in this case, these 3 center pivots, what we're looking at is that phosphorus solubilization index, the same one we were just looking at in the last couple case studies. And we can see this top field is scored at 55, is pretty high. It's at least medium as far as phosphorus solubilizers. But these bottom 2 fields, especially the center pivot on the right, scored less than half that, 24. So that's in the low ranges of our phosphorus solubilization index. So this is where you can pinpoint where to apply those products like Spectrum and others that can help aid in inoculating phosphorus solubilizers. And you can even, on a precision ag level, like this field here in the bottom, we see some blue on the west side, but in the southeast, a lot of yellow. So you can even use this to precision apply biologicals where they're needed and really pinpoint the issues rather than just a blanket solution or a guessing that, okay, you may need this product, so let's just apply a biostimulant across the whole field. So really with BeCrop Farm, what we're doing is bringing precision ag into the biostimulant space and biology space. So very powerful tool that kind of follows up on the precision ag tools that have been used for fertility that have gained a lot of popularity in the last 10 or 20 years or so. So with that being said, there's even some materials here we wanted to get to, but I know we're really up on time, so I'm going to try to ask a few questions here in the last couple minutes we have left. One question we got from the audience is, how is resilience measured in this context? Are we specifically evaluating functional redundancy, or are there other metrics that capture resilience more comprehensively? So that's a good way of, functional redundancy is one way of looking at it. I look at it like adaptability, and that's, I think, Steve, you had a really good analogy there. If you don't have a good backup quarterback in football and your quarterback gets knocked out, then you're often in trouble. You don't have a very resilient team if your backup is a rookie and inexperienced and doesn't have great chemistry with the starters. Functional redundancy is an important component of it, but we're not just necessarily looking at, okay, this is more redundancy is more resilient. We're also looking at how those ecological roles complement each other. So if we have microbes that synergize, that work really well together, microbes, they aren't just doing their own thing in the soil, they're cooperating or they're antagonizing and competing. So that's what the resilience also takes into account. If you have more microbes that, if they're under stress and the microbes' functions can complement each other, that may score better. If you have microbes that are more kind of antagonistic, and I like to use analogies of like cities. Like if you have a town that has not just one medical center, but has a distributed sort of medical facilities, then if there's a natural disaster like an earthquake, then victims of the earthquake, they can access those medical centers if they're well distributed throughout the city. But if you have a city that just has one big medical complex and there's an earthquake and that medical complex gets cut off, road or transportation goes down, then that city is not as resilient. So there's a lot of different angles we look at resilience. Functional redundancy is part of it, but ecological synergy is another big part of it.
And I think a lot of times that's actually based off of just soil health, plant health, environment, all of those based off of habitat increase that ability to have resilience within that soil environment.
Yeah.
Yeah, very good point.
The plant dictates who's there.
When it can, when we give it the chance to.
Yeah, I know we're at time now, so we're just going to answer one last question. Then any other questions we've received, we'll get back to you guys over email and follow up. So this last question is pretty long-winded, so I'm going to try to really concisely summarize it. I'm trying to improve soil microbiology in my grape vineyard, but find it difficult when dealing with extensive drought, 6 to 8 week drought. I believe it is a waste of time to broadcast spray a microbial fish hydrosylate spray on the soil during a drought. I'm assuming because there's no irrigation, it'll dry out. It makes sense. No way to get biology and nutrients into the soil zone in a drought. Thus, I rely on foliar sprays to help with soil biology, life diversity. Does long-term experience show that foliar sprays may still provide improvements in soil biology during extended droughts? Now, this is a unique scenario of extended droughts, so I can't necessarily speak to someone who's dealing with 6 or 8 weeks of specifically no rainfall, but we have had folks who are looking to evaluate how foliar applied products affect soil biology. And the answer is yes, we have seen evidence. We've even seen some foliar applied biostimulants and complex fertilizers have some benefits in the biology. And I think Dennis and Steve really alluded to the reason, the phenomenon behind this well with the rise of phagocycle and root exudates. If a crop is under a lot of drought stress and the microbes in the soil are dried up, they may be metabolizing a little, but when it's really dry, microbes kind of slow down because they don't have that moisture there. And foliar applied, nutrients or other products, what they can do is they can kickstart, the plant's root exudate output, give the crop the nutrients it needs through the foliar side, and then as the crop is able to, use those nutrients and produce root exudates and carbon in the soil, it may stimulate that biology and help give it at least something to work with. It's hard with drought because that can be a limiting factor in and of itself. But foliars, we don't preach foliars as any kind of like silver bullet end all be all solution, of course, but there is evidence that those foliars can sort of give the plant an ability to kickstart that beneficial cycle and put out root exudates so that the microbes can thus help. We've seen that with our testing, and we've had folks who are skeptical that say, I'm going to test a foliar and I don't expect it to help the biology, but then we see that the biology actually has a significant response after those foliars are applied. It's very important if you're looking to evaluate this to sample in that root zone so that we can best see really, what is the change in the microbiome, where those root exudates are being emitted from the crop root. But I love that question was asked because that really gets to why we look at sort of a holistic solution is instead of segregating the plant, the plant and the soil, think of how those roots in the crop interact with the plant, and that can be an important factor in applying foliars, applying soil applied solutions, applying crop protection products on foliage. But it's all, as I think, Bruce Tainio really provided a lot of enlightening, wisdom on, it's all interconnected. It's all part of a big system. And I think that's, a good note to end on here. I wish we had time for more questions, but like I said, we'll be sure to get to you. I know we're already a few minutes above the hour, so we'll conclude things here, but I think we've had a lot of great material today. I really want to thank you, Steve and Dennis, for joining. I think you guys do so much for the regenerative ag movement and the biological space of being able to provide important, reputable, and well-established biostimulants and biologicals to the marketplace. And really appreciate all the insights you provided here today. Looking forward to further conversations because it's always a good time. I always have a new sort of angle or tidbit to think about soil health from when after hearing you guys, give your wisdom. So thank you for joining, and yeah, thank you all in the audience for tuning in today. Really hope that this was some valuable information, and of course, we'll be following up with those with questions and have our information here on this slide if you'd like to reach out to Dennis and Steve or reach out to us at Biomakers. Thank you all.
Thank you, guys. Thank you, Biomakers. Great opportunity, great information, and really appreciate it. Thank you, everybody.
Yeah, thank you all. Have a great rest of your day. Bye-bye.